Good afternoon. Today's lecture is going to be a continuation on discussing the molecular basis of genetics. Briefly, in our last lecture, we discussed that there is in fact a flow of information that occurs with response to molecular genetics. DNA, as you recall, is in fact the storage molecule that contains all of the information necessary for a cell to survive. Very simply put, DNA contains all of the instructions to synthesize the proteins that a cell needs to survive. And as such, it is very important to have a quick, accurate, and precise mechanism that allows for the copying of the genetic material to take place during the S phase of either mitosis or meiosis. Therefore, our focus today will be on the mechanisms of molecular replication or DNA replication. Now, in our last lecture, we talked about the work of Watson and Crick and how they published a paper that described the structure of the DNA molecule, specifically the double helix shape. It turns out that almost immediately after that paper was published, they published a second paper that contained absolutely no research or data in it. What it did in fact contain was a speculation. And I'm giving you a quote here from that second paper. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism. What they were specifically referring to was the concept of sequence complementarity. And the sequence complementarity, as we discussed in the last lecture, is that the A nucleotides will always hydrogen bond or base pair with T's, and C's will always hydrogen bond or base pair with G's. This is what's referred to, again, as sequence complementarity. The two strands of DNA that make up the double helix are in fact complementary. If you know the sequence of one strand, because of the base pairing rule or sequence complementarity, you automatically know what the sequence of the second strand is. There were in fact three proposed models for the mode of DNA replication. They are referred to as the conservative model, the semi-conservative model, and the dispersive model. Now, Watson and Crick, again, in this paper that was published, did not have any experimental evidence to support their idea, but what they proposed was that DNA replication occurred in a semi-conservative fashion. There were, in fact, very eloquent experiments conducted by a separate set of scientists that did in fact support the idea that semi-conservative replication was the mode in which DNA did in fact replicate. I'm not going to go into detail about those experiments. It's enough to say that there is in fact experimental evidence to support the semi-conservative mode or model as Watson and Crick predicted. So what is actually proposed in the semi-conservative model or mode of replication? The first step is that the two original strands of DNA need to separate. Once you have these two strands separated, because of the sequence complementarity that Watson and Crick proposed, again, if you know the sequence of one strand, you can fill in the information of the missing strands. A always binding to T or T with A, and G always binding to C or C with G. So again, if you could separate the two strands, each one of those strands could act as a template for the synthesis of the missing information. And this is exactly what semi-conservative replication proposes. The two strands separate, each one acts as a template for the synthesis of the missing information. The two DNA double helix molecules that result at the end of a round of replication will contain one original strand and one newly synthesized strand. So in this slide, in the figure where semi-conservative is being denoted, you can see the original strand, the double helix in pink, and at the end of a round of replication, you see two double-stranded DNA molecules, one with a pink strand and one with the blue strand. The pink represents the original DNA, and the blue represents the newly synthesized DNA that was synthesized during that round of replication. The research emphasis quickly moved towards the enzymes that are involved in the DNA replication process. And as it turns out, in 1957, we have the work of Arthur Kornberg and his associates. They were the first to isolate an enzyme in E. coli that directed the synthesis of DNA in vitro, which means in the test tube or in a cell-free system. They named this enzyme DNA Polymerase One because it is an enzyme that makes DNA polymers. Once they isolated this enzyme, they conducted a variety of experiments. What they found from those experiments was that there were some requirements for this enzyme. One of the requirements is that all 4 building blocks are required in order to synthesize DNA. And it turns out that these are deoxynucleoside triphosphates, meaning that the building block required to synthesize DNA is a nucleotide that has 3 phosphate groups linked one right after the other. And you can see this on the very bottom of this figure or slide. They also found that DNA synthesis by DNA polymerase 1 required a template for DNA synthesis to take place. This enzyme could not just synthesize DNA at random, but it needed a template in order to synthesize DNA. And the last major finding from their work was that DNA synthesis always occurs in what's known as the 5' to 3' direction. What that means, if you focus at the bottom of the slide, you can see the incoming nucleotide, which is a nucleoside triphosphate, is entering in and being added on to a growing chain. It is the 5' end of this nucleotide that is getting added on to the 3' end of the growing chain. Shortly after that, 2 additional DNA polymerase enzymes were isolated from E. coli. These were called DNA polymerase 2 and DNA polymerase 3. This table summarizes some of the activity of these enzymes. First, you can see for all 3 enzymes, DNA polymerase 1, 2, and 3, none of these enzymes can initiate DNA synthesis, meaning they cannot just jump right in and synthesize DNA. And we'll get back to that in a little bit. In the next line of the table, you can see there's a check mark next to all 3 of them for 5' to 3' polymerase activity. All 3 of these enzymes do, in fact, synthesize DNA in the 5' to 3' direction. In the next row, you can see 3' to 5' exonuclease activity, and all 3 are checked. An exonuclease is an enzyme that can degrade DNA, specifically off of the end of the molecule. So the 3' to 5' exonuclease activity is essentially what's referred to as the proofreading activity. These enzymes synthesize DNA in the 5' to 3' direction. However, if an incorrect nucleotide is inserted, the enzyme can back up, cut out the incorrect nucleotide, which is the 3' to 5' exonuclease activity, and then continue on forward. Lastly, you can see the 5' to 3' exonuclease activity. This is an exonuclease activity that is specific to DNA polymerase one, and that is the ability to remove nucleotides in the 5' to 3' direction. This particular function is typically utilized in DNA repair, but we will get back to another function in just a little bit. It turns out that each one of the polymerases has a slightly different function typically inside of the cell. I'm going to jump down to DNA polymerase 3. It turns out that DNA polymerase 3 is the main replication enzyme. It is the main enzyme synthesizing DNA during DNA replication. It's adding nucleotide after nucleotide after nucleotide after nucleotide. DNA polymerase 2 is an enzyme that is typically involved in repair mechanisms. And DNA polymerase 1 is involved in what's referred to as primer removal and gap-filling synthesis. And as I mentioned previously, we'll come back to that concept in just a minute. Lastly, I want to mention that in eukaryotes, there are a number of additional polymerases that are listed here in the table that just appeared. This table is not complete, but what I wanted to highlight for you is that there are different polymerases with different functions. What I'm really interested in you learning overall is the prokaryotic version of DNA polymerase 1, 2, and 3, and just know that in eukaryotes it is more complicated or more complex. Okay, let's look at this molecular mechanism of DNA replication in a little more detail. What do we know at this point? We know that DNA synthesis is semi-conservative. We know that it is bidirectional. And we know that there are 2 replication forks, meaning that DNA synthesis goes in opposite directions away from the replication origin. In E. coli, we know that the replication origin is a specific set of sequences that is 245 base pairs or nucleotides long. It is made up of repeating sequences in 9 nucleotide blocks or 13 nucleotide blocks. These 2 sequences are different from one another and are simply referred to as the 9-mer, which contains 9 nucleotides, or the 13-mer, which contains 13 nucleotides. So the first enzyme to bind at the replication origin in E. coli is an enzyme known as DNA-A. DNA-A is a DNA binding protein as well as a protein that has protein-protein interactions. As you see on the figure, there are multiple copies of the enzyme DNA-A represented by the little blue circles, and they are all binding to one another like a cluster of grapes. More specifically, though, these proteins can bind to the specific 9 nucleotide sequence in the 9-mer that's part of the replication origin. It binds to that sequence and begins to form a structure that causes a bend or a kink in the DNA chain. The 13-mer sequence is AT-rich. The significance behind this is that A's and T's only have 2 hydrogen bonds holding them together as complementary sequences compared to G's and C's. So there is a tighter interaction between G's and C's compared to A's and T's. So in this case, the 13-mer sequence is AT-rich, which means that when the DNA-A protein binds, it causes a bend at the replication origin. This causes tension and forces the AT-rich region to pop open. This popped open portion is referred to as a replication bubble. The next enzyme to come into play is a molecule that is composed of 2 distinct proteins known as DNA-B and DNA-C. Together, these enzymes act as a helicase. A helicase is an enzyme that has the ability to unwind the double helix. As you can see, two groupings of DNA-B and DNA-C associate with the replication bubble on either side of the opening. This enzyme begins to unwind more of the double helix, making a much larger replication bubble. Not pictured here is another enzyme known as DNA gyrase, also known as topoisomerase. This enzyme also joins the complex on either end of the opening, and its job is to unwind the helix a little bit more, specifically relieving the tension that has been created. The tension by unwinding the helix by helicase can cause DNA damage, and gyrase essentially makes a single cut and allows for a swivel point to release that tension. Lastly, not also included in the figure is a protein known as single-stranded binding protein. The SSBPs are proteins that do exactly what their name implies, bind to single-stranded DNA. By binding to the single-stranded DNA, it prevents closure of that replication bubble so that replication can in fact proceed. Now, as I mentioned previously, none of the DNA polymerases have the ability to just recognize a single-stranded template and begin synthesizing DNA. All polymerases require an attachment point to begin the synthesis process. The attachment point is provided by an enzyme known as primase. Primase is an enzyme that synthesizes RNA, and it synthesizes what's known as the RNA primer. The RNA primer is a short sequence of RNA that then acts as an attachment point for DNA polymerase 3 to jump on and begin adding nucleotides one right after the other. In this figure, you can see the original DNA template strand in pink. You see the RNA primer that has been synthesized by primase in orange, and then the newly synthesized DNA in blue. As I mentioned in our previous lecture, the two strands of the double helix are anti-parallel. That means they are going in opposite directions of one another. Because the strands are anti-parallel, this provides an interesting problem for the enzymes involved in replication. So let's take a look at this figure a little more closely. This figure that I've just added in is meant to be a time progression during replication. The top portion of the panel is earlier in replication, the middle one a little bit later, and then the last part a little bit later than that. So again, time progression, replication moving towards the right of the slide. This allows us to see the structure or location known as the replication fork. The replication fork is right at the point where the enzymes are actively replicating. Now, the top strand, you can see, has the 5' to 3' direction with the 5' end on the left-hand side and the 3' end on the right-hand side. You can see that DNA synthesis on this strand begins at the replication fork. The orange again denotes the primer, and then the blue represents the DNA synthesis. On the bottom strand, you see a similar situation. Because the strands are anti-parallel, the bottom strand is 3' at the left-hand side and 5' on the right-hand side. DNA synthesis always occurs in the 5' to 3' direction. So on the bottom strand, once DNA synthesis is initiated, as long as we keep unwinding DNA, DNA synthesis will continue occurring without stopping. This is referred to as continuous synthesis. However, on the top strand, because synthesis must always occur at the 5' to 3' direction, and the enzymes are always at the replication fork, DNA synthesis is going in a direction away from the replication fork. This presents a problem overall, and that problem is solved by discontinuous synthesis. On this top strand, DNA synthesis occurs in short bursts, representing the discontinuous DNA synthesis. So, continuous DNA synthesis, not a conceptual problem. As long as we keep unwinding DNA, DNA synthesis can continue. The strand that continuous DNA synthesis occurs on is known as the leading strand. The top strand in the figure, again, represents discontinuous synthesis, DNA synthesis going in the opposite direction of the enzymes. This occurs what's known on the lagging strand. Now this figure sort of puts all of the puzzle pieces together. I want you to focus on the bottom portion of the panel, not the top portion. But on the bottom portion, you can see where the original double-stranded DNA is that is undergoing replication in pink. That's the right-hand half of that bottom panel. And then to the left, you see the DNA synthesis taking place. Again, pink representing original template, blue representing newly synthesized DNA, and the enzymes that are all involved in this process. DNA polymerase 3, single-stranded binding proteins, preventing closure, helicase, unwinding the DNA, primase, synthesizing the primer, DNA gyrase, or also known as topoisomerase, relieving tension, and hence the process continues. In Blackboard, I've also provided a link to a YouTube video that really nicely shows the replication process taking place. I strongly suggest that you take a look at that animation so that you can see this in motion rather than just in still slides. This process continues until all of the DNA has been replicated. Some of the finishing steps that are required is to remove the RNA primer. So a molecule that is a hybrid between RNA and DNA is actually very much less stable than a DNA-DNA molecule. So all of those primers that have been inserted by necessity to get DNA replication to take place have to be removed and filled in with DNA. The main enzyme that is involved in doing this is DNA Polymerase One. Like I said, we would come back to DNA Polymerase One towards the end of the lecture, and this is where DNA Polymerase One typically fits in. Its job is to cut out the RNA primer and fill in the gap with DNA. The very last enzyme is an enzyme known as ligase, and ligase just closes up the covalent bond that has been missing once the primer has been removed. So ligase closes up the covalent bond, and now you have two seamless strands of DNA. So typically with most of my molecular genetics lectures, I focus on prokaryotic models just so that we can get a good understanding or idea of what's happening with the mechanisms. Eukaryotes tend to make things much more complicated because there are a lot of additional regulatory steps involved. I do want to highlight a few things with eukaryotes. That is, DNA replication does also begin at specific location known as the replication origin. Now, the sequence of the replication origin is different than that in prokaryotes, but there is a specific set of sequences where replication begins, known as the replication origin. DNA synthesis also occurs bidirectionally in eukaryotes, meaning that once replication has been originated, DNA replication moves in either direction away from that replication origin. However, one additional problem that comes into play is that eukaryotes typically have much more DNA compared to a prokaryote. Yeast, which is a single-celled eukaryotic organism, has approximately 4 times the amount of DNA compared to E. coli, whereas Drosophila has something like 10 times the amount of DNA. This presents a very big problem in getting DNA replicated quickly and precisely. Most importantly, quickly. The solution in eukaryotes is to have multiple replication origins. In mammalian cells, there are or can be up to 25,000 replication origins.